Morning. My name is Randall Kaye. I'm the Chief Medical Officer of Longboard. I've been a sort of interesting background history. I'm a pediatrician and an immunologist by training, and I've been in drug development for about 30 years. Successfully had about 5 medications approved and probably studied well over 50. And those are actually probably good numbers statistically. So it's been a pretty exciting journey so far. But I think I'm supposed to start with the fun fact. So, last night I went to an Orioles game. I looked out the back window of the Hilton and there's Orioles Park. And it was empty. You know, best team in, in baseball right now. What's that? Okay.
We're playing the Rays, and we're 2 ahead of them.
It is the Rays, and it's a really important game. So I got that. So we got in, we walked in like the 3rd or 4th inning, Michelle, it's still pretty empty there. So I was sitting there, and it's like, great game, and I look right by home plate, and there's a pitch clock. And I'm thinking, I wonder if that's distracting at all, if that I know it speeds up the game and stuff. But I was like, yeah, it's probably not so bad. And now I'm looking that I have a pitch clock, so I'm gonna get started. So today you're going to have the opportunity, whether you like it or not, is to peer into the brain of someone who works in industry and to see how we kind of think and approach things. But I have sort of a little bit of a question for you. When you're a drug developer, how do you know that your compound is a drug? When do you know it's oh, I'm really working on a drug? And there's not a right answer from an FDA standpoint, but I'll just give you our view. It's when you're doing your first in human studies, because everything up to that, it's just a compound, it's just a molecule. It's something that you're working on that you think might have some interesting promise. Now, some of us in drug development think about the drugs that we develop as our babies, and they are. And we want them to do really, really wonderful and really great. And I'll touch upon that in a moment. So how do you know when you're a baby, when the medication you've been working on has grown up? And the way we simply look at it is it's when your medication is approved and it's able to be utilized across the board. And it's a little bit of a fundamental difference in the way that we might think in industry is our goal is really a little bit more around approval, not so much sometimes on how things are actually utilized. So this slide is just to give you a little bit of a sense of sort of the generic ways of sort of looking at things before we start a trial. Obviously, we want to measure outcomes of clinical trials for conditions involving seizures and non-seizure outcomes, and it requires a lot of careful thinking and planning. And our goal, obviously, is to capture the impact of the treatment being tested both on the frequency and severity of the seizures, as well as any other outcome. Now, we have this concept in drug development called target product profile. We want to think about how we want our drug to perform later, even though we don't really know how it's going to perform a priori. So we're putting this kind of promise together. It's kind of like when I was born and my mom looked down at me and said, he's going to be a doctor at hour 1, and assuming that was going to be true because she said it. So that is sort of the way we kind of think of our compounds, is that they're going to grow up, they're going to do really, really well, and they're going to meet all these promises. And then on the right-hand side of the slide, here's our sort of list of how we think of things. But first and foremost, we have to look at sort of background precedent of what are the primary outcomes that are typically seen in this area, and it's seizure frequency. And there's a whole long list of secondary outcomes that you see here that we start to think about and then wrestle with that concept of, are we going to look at objective measures or subjective measures that are more patient-reported? Are there some standardized assessment tools that are out and available? Are we interested in long-term versus more short-term? What's going to be more important at a patient level? What's the benefit-risk ratio? It seems like we're going to get great efficacy from this medication, but maybe we're going to have some limitations due to its adverse event profile. And then we have to, of course, apply some statistical analysis to make sure whatever we're promising in our primary outcomes actually is statistically significant, obviously clinically meaningful. But it also goes for our secondary outcomes too. If it's not statistically significant, it's unlikely we'll ever be able to talk about this. So I'm going to give you 2 kind of brief case studies. One is from a medication that Longboard that we're currently working on, and the other is from a case study of a medication that I worked on about 10 years or so ago. So LP352, our baby, we think it has the potential of making a difference across a broad range of DEs. There's a couple things that we know about it or knew about a priori. Has greater selectivity, specificity as far as interacting with the 5H2 receptors. We know it penetrates the brain in a dose-dependent manner. There's some early proof of concept observations with other compounds. Fintepla probably is a good example that well interacts with the 5-HT2C receptor. But our compounds, we think, might be a better approach because it's only interacting with one of the receptors. We have predictive efficacy because we use some non-clinical models to help us think through it a little bit. And then it became a drug by going into first-in-human studies where we were able to do a series in healthy volunteers of looking at how it behaved to see if they might have some potential biomarkers that we could utilize, and then understanding the pharmacokinetics and pharmacodynamics of the compound. And then we start to march into our controlled clinical trial, which I'll talk about in a moment. But before we get started on the controlled clinical trial, we're like, well, we know there's precedent. We know the summary of the studies. Dr. DeLugos did a great review of the medications that are currently available. So we do what every other drug company does, is we look at precedent. And precedent is, and I apologize if the numbers aren't perfect here in terms of the prevalence in the U.S. for certain conditions, but the often typical drug development approach is study one condition, move on to the next one, move on to the next one. Start with Dravet, maybe start with Lennox-Gastaut, maybe move to CDKL5 or tuberous sclerosis. We think that's an interesting approach, but if you look at the right-hand side of the screen, we kind of think it's interesting. What about all the other DEs? Will they ever even get studied? Or are we going to leave the families with medications that never ever have safety and efficacy information? So we were wondering, maybe we could consider studying all of them at the same time. And so when we look at the data for primary outcomes, the one thing that's going to facilitate getting your medication approved is were these seizure outcomes? What was the general percent decrease in seizure frequency? And as you look over the right-hand side of the screen for fenfluramine in Lennox-Gastaut, lacosarin, ganoxolone, the cannabidiol, and so forth, you see very sort of similar response rates. And we also know by precedent is that clinicians will use anti-epileptic medications or anti-seizure medications fairly broadly. So our thought was, at least as we're getting into a proof of concept, why don't we do an all-comer study? Why don't we try and see what we learn? So when we think about, and I don't mean to make it sound pejorative, but when we really do think about our baby, we don't know how it's going to perform. We just know how it should by precedent and the receptors that it's interacting with. So we use our initial data to help give us some hints on what the right dosing should be. Dennis did a nice review of the structure of the clinical trial designs. This replicates exactly how people are generally approaching it. But then comes the part about outcomes, seizure versus non-seizure outcomes. This is a proof of concept study. So we actually have to start with safety and tolerability. That's first and foremost. And then our most important efficacy outcome, it has to be reductions in seizure frequency. It's it. If we don't study that first, we don't know if we have a drug that might get approved someday. It's a critically important test. Fortunately, we just completed enrollment. We're looking towards having top-line data in this study in 50 patients with a variety of DEs around the end of the year. And I can tell you that there is a large number of patients that have LGS as a baseline DE. So we're really excited to be able to see, can you actually treat patients across the board and have similarities in their seizure outcomes? I'll give you another quick case study. Pseudobulbar affect. Tracy looked at my slides, sent me a note. It's like, I'm not getting it. I'm not sure. And then we agreed, right? It might work. So what is PBA or pseudobulbar affect? Neurologic disorder, causes involuntary emotional outbursts, crying, laughing, or other emotional displays. They're incongruent or excessive or exaggerated relative to the patient's inner mood. They occur several times a day. The episodes last from seconds to minutes. They're pretty sudden, and they cause pretty severe impairment. Now, I don't mean to make this sound pejorative, but when we were first working on the development of this, people used to refer to PBA attacks as emotional seizures. It's actually exactly what it looks like. And that's why I thought it'd be sort of an interesting case to put in front of folks. So in patients with PBA, they typically have an underlying condition, ALS, MS, traumatic brain injury. And so you would think, is PBA really the most, the most problematic thing that these patients are actually having and are challenged with? In many ways, as it relates to relationships, the ALS is one aspect of their condition, but this is just a quote from one woman. I've had ALS for 7 years, and this emotional problem is just as debilitating to me as the paralysis. So that was the program that we had embarked. It was called the STAR trial. It was a compound called Lennox-Gastaut. I'll tell you the end of the story. The medication was subsequently approved. It's broadly utilized. But here's the challenge that we faced. We had to do a primary endpoint that was going to be acceptable and measurable from a regulatory and a clinical standpoint. So the one we ended up agreeing on was counting laughing and crying episodes from the patient diary. The first study that we conducted actually used a validated scale. It's called the CNS-LS score, which is very well recognized in the community. When we were talking with FDA, they were like, well, it's a validated score. I'm not really sure that's going to be the best way of approaching it. Tell us why you want to use the CNS-LS. Like, well, it broadly gives us much more insight than just counting the seizures, because it gives us an impact of how it's having an impact on patients and families. And so they said, well, what is it validated against? And I said, laughing and crying episodes. And they said, okay, so kind of think maybe you should be using laughing and crying episodes as your primary endpoint. So we had to make a little shift and a bit of a pivot. And so the primary analysis, I'm not even going to read this to you because I think the way we wrote it is great from a statistical standpoint, great to prove that there is an impact on the drug, but it is uninterpretable from a patient or a family standpoint. The secondary endpoints are actually rather interesting, because the secondary endpoints gives a little bit more insight into not just the concept of can you move forward in getting this medication approved, but more importantly, will it ever get utilized, which is much more important of whether a drug is on the market. It's actually whether or not it's going to have an impact that people feel is going to be worthwhile to consider the compound. So here's our primary endpoint. It was fun when I was presenting this to investors and to investigators, because they were like, you're using the negative binomial model longitude analysis and your treatment effect is 0.53. It's a great p-value, but what does this actually mean? I'm like, well, the treatment effect above and beyond placebo is 46.9%. And he said, it's cool. So 46.9% reduction? No, it's a negative longitudinal binomial. So it was really hard to explain to people what this really kind of meant. So we had to use a series of secondary endpoints. So I'm just going to fly through this. So some of the secondary endpoints actually are a little bit more helpful and more important. We define patient with remission as having no episodes during the last 2 weeks of the study. So we're able to say about 50% of patients actually reach that outcome, compared to less than a third. That was meaningful to patients. We looked at clinical response, which was defined as a 40% decrease. So this is part of the experiment. We don't know how this compound was going to behave. It's okay. Not outcomes that are actually all that exciting. Percent of episode-free days, very, very important from a patient standpoint. Just give me a sense of how many days I'm not going to have to go through some of the challenges that I'm facing with my mom or my dad or my sibling. Then we did a series of other secondary endpoints. We looked at that CNS-LS score, and as expected, really robust findings. We spent more time after the medication got approved talking more about the scale, because it was a broad-based scale and it was more meaningful to patients than just how many less seizures am I going to have. And we took a shot at a number of other secondary endpoints, and we did them in order. We looked at the NPI, which is a neuropsych inventory, Beck depression, that we saw some inklings of some impacts. So now we could take this data and say, let's kind of think a little bit about some additional studies we may want to conduct post-marketing to give some better understanding of how the medication could be utilized. We looked at health outcome scale, looking at the SF-36, purely to give us some hints of what should we be looking at further post-marketing studies to help people understand what the potential impact is. And it's the biggest challenge that you face in drug development is you don't know how your medication is necessarily going to perform until you run the experiment. And so I'm sometimes challenged when I hear people say, but seizures and drops in seizures isn't the most important thing. It's like, I know, I get that, but unless we focus on that at first, we're not going to have a medication that's going to get approved. So on the right-hand side, this is the peer into how we think. Our primary objective is utilizing primary outcomes that support the drug's approval. That's square one. And we all know, especially those of us who are clinicians, the secondary objectives are way more important as we utilize these secondary outcomes that help understand how a drug will really be utilized by patients and families who are trying to manage in this journey of living with their disease. Thanks for listening.